An article published in the American Physiological Society journal in 2016 discussed insulin allergy in the brain and the effects thereof. The study behind this article was done by researchers in Tübingen, Germany. A second article, published in the Frontiers in Science journal in 2019, outlines the beneficial properties of insulin and neurodegeneration from a lack of. This study was done by researchers in Naples, Italy. Insulin resistance in the brain is found to be caused by problems with weight, age, dementia, and type 2 diabetes. Researchers in Brussels, Belgium, have estimated type 2 diabetes cases to reach nearly 600 million in the next 14 years. It was in 1927 when insulin was discovered and was later found to be necessary in brain tissue function. Connective tissue in the brain depends on insulin to turn on glucose transporters for circulation of glucose. Documented results of brain insulin activity were found to have had an effect on behavior and metabolism, specifically on cognition in both human and animals. As seen on Figure 1, there are several cognitive and metabolic issues connected to insulin resistance in the brain. Specifically, a study found overweight expectant mothers as a cause to insulin resistance in the hypothalamus in fetuses. Figure 2 highlights all regions of the brain affected by Alzheimer's disease due to insulin resistance. Alzheimer's patients are found to have elevated levels of plasma insulin, while at the same time show lower insulin resistances. Because of this, reduced uptake of insulin leads to slower insulin actions. Specifically, 2 studies were done on how diabetes impaired neurocognitive functions. The longitudinal study, done over a 6-year period on middle-aged to older adults, found those with type 2 diabetes had done poorly on nearly all neurophysiological tests. The most significant decrease in test scores were from categories on memory, information processing, and mental skills. The cross-sectional study found similar results on tests for physical motor movements, verbal communication, and learning ability from the type 2 diabetic patients. Brain atrophy has been linked to obesity. Brain atrophy is a loss or shrinkage of brain tissue which carries a higher risk of strokes or brain bleeds. Advanced magnetic resonance imaging has detected smaller gray and white brain matter in patients of obesity and type 2 diabetes. Alzheimer's patients are known to have issues with amyloid fibrils, which is a form of plaque that builds up between nerve cells and neural pathways of the brain. Functional connectivity is also compromised, in which regions of the brain have issues interacting one with the other. Insulin is a hormone that is released from the pancreas when glucose levels rise from either drinking or eating substances that contain sugar. Insulin is also found in different areas of the brain, such as the hypothalamus, olfactory region, which is responsible for smell, and the midbrain, which is at the very top of the brain stem. It is still yet unknown if insulin is produced in the central nervous system. Research has found that insulin transmission to the central nervous system is decreased due to obesity and hyperglycemia. Another beneficial property of insulin is its role in neural cell development and endurance and the ability to protect developing fetal retinal cells. This figure is more or less a blueprint of results found during an experiment on the modulation of the alpha synuclein expression and aggregation. It shows where insulin signaling in the brain help regulate neural activity by using receptors to pass through the intracellular enzymes that are essential for the activation of the PI3K and the AKT pathway. This process hinders the substrates, which reduces aggregation or the formation of clusters and ensures survival of neural cells. Insulin is also vital in the management of mitochondrial function, which gets and stores energy in the respiratory chain of the brain.